Hello, my name is Cameron Herman, and today we are going to talk about microscopic monsters called viruses. What are viruses? Are they even alive? They technically are little packages of genetic material, so they aren't really alive until they come in contact with another form of life, usually a bacterial cell. As I mentioned before, viruses are little packages of genetic material. Inside the packages, there's either double-stranded RNA or DNA or single-stranded RNA or DNA, depending on the virus. Surrounding the genetic material is a protein coat called a capsid that acts as a shell around the genetic material. Viruses can be split into two groups, enveloped and non-enveloped. The envelope is a lipid membrane that surrounds the capsid and helps transfer the viral genetic material from one cell to another. The envelope is made up of pieces of other cells that the virus has already lysed. Working with the glycoprotein spikes, envelopes act as a type of Trojan horse for the viral genetic material by disguising it to look familiar to the cell. The cell then either lets the genetic material in by fusing the viral envelope to the nuclear envelope or by eating the whole genetic package in a process called endocytosis. Then the life cycle starts over. The life cycle begins when a phage or little package of virus attaches and injects its genetic material into a host bacterial cell. The genetic information then inserts itself into the DNA of the bacteria in the form of a prophage. The viral DNA is then replicated with the DNA of the bacteria, which codes for the formation of more of the virus. The virus that was coded for begins to synthesize within the bacteria, creating the empty phages. The phages are then packaged with DNA. Tails are finally added and the phage is completed. The bacterial envelope erupts as the cell is lysed and all of the new baby phages are released. This is called the lytic cycle. Viruses can also undergo a lysogenic cycle. It begins when viral genetic material is inserted into the bacteria and the viral DNA inserts itself into the genetic material of the bacteria. When the cell prepares to split, it copies its own DNA, including the viral DNA. This creates two copies of the DNA. When the cell splits, both of the daughter cells contain viral DNA, and the lytic cycle can continue. Because viruses are so infectious and spread so easily, they can be very dangerous. Some examples of viral infections that you would recognize are the influenza, herpes, HIV, and polio. Remember, despite this, you can avoid catching a virus by using soap and water to wash your hands before touching your eyes, mouth, or nose, and practicing good hygiene. Thank you.